So we've looked at double-stranded DNA structure. We've looked at chromatin structure. It is the complexing with histones to make the nucleosomes and the 30 nanometer fiber and so on. We've looked at the chromosomes and some of the elements that are required for their stability and DNA replication and telomeres, centromeres, and so on. And what I want to start doing now is looking at genes and gene expression. There's a short reading down in the bottom, 228 to 238. So, like I said, you don't have to read everything in the text, but these are regions of the text that really point to what we're talking about. So, let me just talk a little bit about the genes and which genes we're going to be looking at in particular. So, on the left-hand side, I have a little table that I wrote out. Again, we're going to be looking at gene structure, expression, the structure of what a gene looks like, gene expression, control of that expression. And the genes that we're going to be looking at are genes that encode proteins. And of course, those proteins give rise to function. And also, we're going to be looking at the genes that encode some non-coding RNAs, which also have function in the cell. Now, there are different classes of genes. There's genes that encode ribosomal RNAs. There's lots of those. There's genes that encode transfer RNAs. There's genes that encode, like let's say the small microRNAs and other RNAs that are involved in regulating the cell and gene expression. But we're really not going to be looking at those. We're going to look at a particular class of genes, and those are the genes that basically encode proteins. Because the proteins are what we've been looking at, and the proteins carry out so many functions. And also we know most about those genes because those are the ones that have been studied most extensively. So if we're looking at protein coding genes and we look at a eukaryotic cell like a human cell, really only makes up around 2% of the genome. 98% of the genome is not encoding protein. So we're really only looking at a very small part of the genome. That's just that 2%. The other 98% encodes other things like I mentioned, the ribosomal RNAs and transfer RNAs, but also there's lots of other structures and repetitive elements in the DNA, and maybe we'll have a chance to look at some of that later. But right now we're just looking at these 2% that encode proteins. So I just wanted to give you a range of how many genes, protein encoding genes, exist in different kinds of organisms and viruses and so on, just so you can see what we're talking about, what we're looking at. So the smallest are obviously the viruses. And here's a virus called Phi-X174. It's a bacterial virus, just 11 genes. A virus like this enters a cell, and then some of those 11 genes, the proteins take control of DNA replication and protein synthesis and transcription, and they take over the cell and then produce more virus. Epstein-Barr virus is a human herpes virus. This is actually the virus that causes infectious mononucleosis. It also is related to some cancers. And there's 80 genes, so it's a much bigger virus. The Mycoplasma is kind of the smallest free-living organism that we're aware of, and they have 680 genes. E. coli is the bacteria, 4,408, and then it goes up. Yeast around 6,500, then Drosophila, Arabidopsis, and then humans. It's estimated that we have around 30,000 genes, 30,000 protein coding genes. Now, if you look on the right-hand side, this just gives a little bit of an outline of the process, and we've seen this before. So you have the double-stranded DNA. A region of that DNA is the gene, or a gene that will encode protein. And again, in humans, it's only 2%, so it's a very small amount. And then it shows RNA synthesis or transcription. So that's the first step. When we looked at the information flow, we kind of talked about that. The DNA opens up, and the RNA polymerase comes in, and then it transcribes the RNA. There's you have the RNA there, and then it goes on to protein synthesis through translation. So we're going to talk about each one of these things, but today we're going to be talking about really not so much the RNA synthesis, because there's not a whole lot to say about that. We know that the nucleotides add on to the 3' hydroxyl, and they use the template in order to decide which base to put down, but we're going to be looking at transcription, and particularly transcription regulation. So the next slide I'm going to show you is one of my drawings, and there's nothing quite like this in the book. It's a little bit, hope you'll be able to read my writing, but we'll go through it slowly, and it looks pretty busy. So, but I like it. So here it is. So this is basically a drawing showing the general architecture of a eukaryotic gene. And we'll walk through this. So if you look at the very top, there is a kind of a rectangle. This represents double-stranded DNA, basically. But the very top represents, where we have the rectangle with those boxes, represents the transcribed region of the gene. So there's a very specific region that's transcribed into RNA. It's not a huge, long, half the chromosome. It's very specific. In fact, there's a very specific start site, which is shown by that arrow. So the RNA polymerase knows precisely where to begin transcription, and that's called the +1. There's a numbering system, which I'll talk about in a minute, but the +1 represents the first nucleotide that is synthesized. That's what is represented by the arrow, which represents the transcription initiation site. Then you look down that rectangular box, and at the end there are termination signals. So it also knows where to start, and knows where to stop. So when transcription occurs, the RNA polymerase comes in, and the arrow down shows transcription, and you get that primary transcript, which has the 5' end and the 3' end. And that's primary transcript. It's not capable of translating into protein because it has the exons and the introns in there. The exons contain the coding sequences. The introns are non-coding, and the introns need to be removed. And we'll talk about splicing later. It's quite sophisticated, quite complicated, but we'll talk about it later. But in any case, the coding sequences for the protein are split up into those regions. So that's what the hatched boxes represent. And then ultimately, the primary transcript is modified. There's a 5' cap, which we'll look at later, polyadenylation, that run of A's at the end, and then, of course, splicing, which takes out the introns, the non-coding introns, and moves the exons together, so you have what's called an open reading frame, and it contains the codons for the protein. And then it gets translated to the ribosome. But we're looking really up at the top. And so you have the transcribed region, and then there's what are called upstream sequences. And there are downstream sequences. These are DNA sequences. They're not part of the transcribed region, but they're regulatory, meaning that they control whether or not the gene will be transcribed into RNA. And then the downstream sequences are also regulatory. They participate in termination, but also they can regulate events up at the top, around the transcription site, by binding to certain proteins. So there's regulatory sequences upstream and what we call downstream. Now there's a numbering system, and anything upstream, which means to the left of the transcription start site, have negative numbers. So you get a number -10, -25, -30 means 30 nucleotides upstream of the start site. And then you can have plus numbers, which begins at +1, which is the first nucleotide that is synthesized, or the position of the first nucleotide, and then it goes downstream. And so you can have, +1, +100, +300, depending on how long the transcript is. And that gives you an idea of where you are in the gene, the numbering. Now, what I want to focus on first is what's called the promoter, and I have that highlighted in yellow. So there's a region right upstream of the transcription initiation site, +1, which is referred to as the promoter. And simply put, this is the region that will bind to the RNA polymerase. And so we're going to focus on that and look at how that participates in regulating transcription. For this example, starting out relatively simple in a sense, we're looking at E. coli bacteria. And we will soon move to the eukaryotic promoter, which turns out to be much more complicated. So we're looking at an E. coli gene that would encode protein. And I want to just mention what's the function of the promoter. This is actually true in E. coli and eukaryotic cells. So what's the function of the promoter? It has a number of functions. It's a specific DNA sequence, and it's recognized by the RNA polymerase, and it signals where to begin transcription. So it identifies the target for transcription. If you look at a eukaryotic cell, a human cell, where only 2% encodes proteins, and there's 98% that does not, then there has to be a way for the proteins and mechanisms to identify where that 2% is. And not all genes are expressed at all times, so there has to be some specificity there, so the polymerase is not transcribing every gene and every DNA sequence, so there has to be some way of identifying what gene needs to be transcribed. That's one of the functions of the promoter. It orients the polymerase, because the polymerase binds in a very specific direction, orientation. So when it starts making the RNA, it makes the correct RNA. If it was in the opposite orientation, let's say flipped, it would go the other direction, which would be wrong. And it positions the polymerase for correct initiation. As I mentioned, that there's the +1 site, the transcription initiation site, that's quite specific. So it knows where to begin transcription. Now, if you take E. coli DNA, that's what's shown on the bottom, the 2 strands, 5', 3', so on, anti-parallel, just any random piece of DNA. If you take E. coli DNA and you take pure RNA polymerase, that is, you purify it from the cell, and there are these assays which are called DNA binding assays, again, which I'll talk about a little bit later. But if you take the RNA polymerase and you mix it with the DNA, it binds to the DNA. And if you sequence that or kind of characterize the region, what you see is that it's not binding to any random piece of DNA. It's binding right over the transcription start site for the gene. So that's what's kind of illustrated there. The +1 with the arrow, the RNA polymerase binds right there. And it covers a region of around 60 nucleotides. 20 nucleotides upstream and 40 nucleotides downstream, so it's a large protein. And then it will begin transcription. If you add the nucleotides, the ribonucleotides, it'll begin transcription and it will transcribe precisely what it's supposed to do, beginning at the +1. So what is special about this DNA sequence that it would allow the polymerase to identify it and bind right there? So people have taken the way this has worked. The way it worked was when this was first discovered, different labs, different scientists took hundreds of these regions that the polymerase was binding to and then sequenced them to find out, is there anything specific or anything unusual about these sequences? And in fact, there was. That's shown over on the right-hand side. So it turns out that there are highly conserved sequences at the region where the polymerase binds. And there's two clusters of sequences that are important. And that's what's shown in that diagram. So they found out that at -10, meaning 10 nucleotides upstream of the start site, there was TAT, ATT, and that's shown in both the, below my drawing and the one below. TAT, ATT was always present at this -10 region. And then there's another region that's important at -35. And there at -35 was TTG, ACA. So all the nucleotides surrounding these regions, like the nucleotides between +1 and -10, or between that region, can be any nucleotides, and then the distance, the region between the -10 and -35 could be any nucleotides, and then of course upstream any nucleotides. So this specific region and also the specific distances between them, turns out that is the specific recognition site that the RNA polymerase binds to. So the binding to this region right there at the -10, -35 positions the polymerase precisely where it needs to be relative to the start site. Of course, there's an orientation there because you have the -10, -35 are different sequences. So the polymerase binds in a specific way on the DNA and then can initiate transcription. So the promoter, in this case, has a very specific DNA sequence that the polymerase recognizes. Now, the polymerase is made up of a number of proteins, and we're going to look at that now. So on the left-hand side, so the E. coli polymerase, there's actually 5 subunits. So there's 5 proteins that are involved, are required for the polymerase. There are, and I have that in brackets, there's one copy of what's called an alpha subunit, 350 amino acids. There's a beta subunit, which is much larger, 151,000 amino acids. And then there's the beta prime, which is 156,000 amino acids. Now these things come together and form a complex, which is called the core polymerase. And then there's a variable factor, which is called a sigma factor. And there's a number of different sigma factors, which I'll mention in a second. But you have the core polymerase, and then with the addition of the sigma factor is called the holoenzyme, the RNA polymerase holoenzyme. Now, if you take the core polymerase, and this is shown below, if you take the core polymerase without the sigma factor, and you bind that to DNA, it will not bind specifically to that -10, -35 region. It'll bind to any DNA, and it will begin transcription making RNA. It'll bind to human DNA, it'll bind to E. coli DNA, it'll bind to whatever synthetic DNA, it doesn't matter. It'll just bind to DNA and start making RNA. When you add the sigma factor to that, it alters the structure of the core polymerase. We consider it an allosteric regulator, so it will alter the structure or the conformation of the core polymerase and cause that now to have specificity, which will then bind to the promoters we were looking at before, the TAT, ATT at -10 and the -35 region. So the sigma factor is a specificity factor. It changes the structure such that it binds specifically to the promoter. And that's just illustrated again on the right-hand side. This is from the book. So you have the start sites. The RNA polymerase is shown in blue. And you have the promoter, which is kind of bracketed out. And the sigma factor is that little yellow ball. So the sigma factor gives it the specificity. Then it goes ahead and starts transcription. And then the sigma factor is released. And you get transcription. But the main point here is that the sigma factor is required for specificity. Now, it turns out that there are, let's see, turns out there are around seven different sigma factors. The sigma factor here that we're looking at is called sigma 70, which is the most common one. For most of the genes that are transcribed use this sigma 70. But there are a number of other sigma factors, and that's shown here. So there are multiple sigma factors that have been identified. So let's start out, first of all, by looking at the column that says sigma. So these are the different sigma factors that have been identified, and the number represents the size. So 70 kilodaltons, 54 kilodaltons. So we have sigma 70, sigma 54, sigma 20, 32, sigma 28, 38, 24, and so on. Now, when these sigma factors bind to the polymerase, they cause the core polymerase, they cause this change that allows it to be specific. So sigma 70 is the one we looked at before. When it binds to the core polymerase, it binds to what's shown on the right-hand side, so that's the promoter with the -10 and -35 region, and it binds to that sequence that we had looked at -10, TAT, ATT, and at -35, the TTG, ACA. Turns out that these different sigma factors control the polymerase, the specificity of the polymerase, and allow it to target different classes of genes. Different groups of genes that have that give rise to proteins with specific functions. So, for example, if you look at the second one, there are a number of genes that are involved in nitrogen metabolism. And those are controlled by the sigma 54 binding to the core polymerase. The core polymerase stays the same. When the sigma 54 binds, it still binds to a promoter, but if you look at the promoter sequence, it's different. At -10, there's TTGCA, and -35, there's another sequence. If you look at the next one down, sigma 32 is referred to as a sigma factor that is required for the heat shock response. The heat shock response is a response that allows the bacteria to survive under a little bit higher heat. That it turns on proteins, turns on genes that make proteins that allow it to survive under a little bit higher heat. Like normally the cells will grow at 37 degrees, which is, like body temperature. But if you raise the temperature to 42 degrees, which is higher, then these heat shock genes get turned on. And so sigma 32 appears. It binds to the core polymerase and then binds to the sequences that are shown on the right-hand side. There's still a -10, -35 box, but those sequences, the TAT, ATA, and the one at -35, which is CCCC, those are present upstream of genes that encode the heat shock response. Then you have one for flagella, starvation, very high heat, iron metabolism. So the sigma factors direct the core polymerase to different classes of genes when the bacteria needs to turn those genes on. Now the sigma 70, as you see, it represents a sigma factor that binds the polymerase, which then expresses what are called the general metabolism genes, general genes. And so those are genes that are required under normal circumstances. And it turns out that probably 80% of the genes in E. coli are represented by that class. So that's why when people purified the DNA that was bound by the polymerase under a normal situation, most of the sequences that identified were the ones that had at -10, the TAT, ATT, and so on. There were a few that didn't, and they didn't really understand what was going on at the time, but the vast majority were the ones that were identified by the sigma 70. So that describes what I wanted to describe about the E. coli promoters. Relatively simple, but you can see the role of the promoter, the role of the polymerase, and how it's a little bit how it's regulated to bind to different genes. Now, when we talk about eukaryotic cells and eukaryotic transcription, turns out to be much more complex in many, many, many levels. There's many more proteins involved. The polymerase itself, whereas the polymerase for E. coli has 5 subunits, that would be the holoenzyme. The RNA polymerase in the eukaryotic cells and in human cells contains at least 15 proteins, maybe more. So everything is more complex. Even the polymerase is more complex. And in addition, there are more RNA polymerases. So initiation of transcription in eukaryotic cells is, like I said, much more complex. There are 3 different polymerases. There's what's called polymerase 1, which transcribes most of the ribosomal RNA genes, so the promoters for these are different than what we're going to be talking about. There's the RNA polymerase 2, which is what we're going to be focusing on, because most of the work that's been done has been done on RNA polymerase 2, because all of the protein coding genes are encoded by RNA polymerase 2. There are some other genes like microRNA genes and other non-coding RNAs which are encoded by RNA polymerase 2, and they'll have similar promoters. And then RNA polymerase 3 encodes smaller RNAs like transfer RNAs, the small ribosomal RNA, and other small RNAs. But we're going to be focusing on RNA polymerase 2 because it is the most well-studied, and also because it encodes all the protein coding genes, and of course those give rise to the most of the functions that are in cells. So we're going to focus in on the RNA polymerase 2. So here's a diagram of what we call the core polymerase. So you have on the left-hand side, the right-hand side, the messenger RNA initiation site with that arrow. That arrow should say +1. That's the first nucleotide that would be synthesized. And then there's also a conserved region, just like there is in the bacteria, but it's at a different position at -30. So it's 30 base pairs upstream of the start site. There's what's called the Tata box. It just refers to it as the Tata box, and below I have the actual sequence, which is TAT-AAAT, is the Tata box. This sequence, the Tata box, is present in probably 95 or maybe higher, let's say 95% of all RNA polymerase 2 transcribed genes. So it's extremely important. In addition, and we'll talk about these later, there are additional upstream, what they call upstream promoter elements that we'll see participate in whether or not the transcription occurs. These are regulatory, but we'll talk about those probably next time. So the Tata box is, if you have a promoter that just has the Tata, not all promoters will have these upstream elements, most do. But you can have a promoter with just the Tata box alone, and that would be referred to as a basal promoter. It's a very basic basal promoter. The Tata box is key because it acts as kind of an initiation site, or a site where proteins bind, that initiates a process which we refer to as the establishment of the transcription initiation complex. It begins by a particular protein binding to the Tata box. Now we're going to look at this figure next, which comes from the book. It's not precisely the way I would draw it, but it's good enough. I mean, it's good, and we'll walk through it kind of slowly. So, as it says, to begin transcription, the RNA polymerase II requires what are called general transcription factors. These are extremely important, and there may be as many as 50 to 75 proteins that bind to this thing, these general transcription factors. And what I've typed at the bottom on the right-hand side, this is the establishment of what's called the transcription initiation complex by general transcription factors. So let's look up at the top. So you have the TATA box, the transcription start site, and the gene. The TATA box is illustrated by that lighter green box. The first thing to happen, and this initiates a sequence of events that occurs, but the first thing that happens is the binding of a complex which is known as TF2D. TF2D is an extremely essential component of this transcription initiation complex because it binds to the TATA box, and then to that other proteins bind. Now, the TF2D is, as I've written there, is a complex itself of as many as 15 proteins. So these are large complexes. You see, it's very different than E. coli. So the TF2D is a complex of a bunch of proteins, and there's two classes of proteins in the complex. One class is called TBP. There's actually a dimer, two proteins together. TBP stands for the TATA binding protein. And actually, if you look at the TF2D up there, you can see that the TBP is shown a little bit darker green, and that is the complex, part of the complex that actually binds to the TATA box. So this is a DNA binding protein that recognizes that sequence. So you have TBP, which is a TATA binding protein, plus all these other factors, could be 10 or 15 factors, which are called TAFs. And the TAFs stand for TATA binding protein associated factors. Now the TAFs are part of this TF2D, and what I will say is that the TAFs can vary. The TBP is always there, but the TAFs can vary a bit, and we'll see that later. So you have the TF2D binds, and then it initiates this sequence of events that other proteins then bind. So then you have the binding of TF2B binds to this site. Then you have other proteins called TF2A, but it's not actually showing TF2A. But what it is showing is below, so you have A, B, C. If you look at C to D, you have the binding of TF2E, TF2H, all these other factors. It even has just other factors. And only then does the RNA polymerase bind to the DNA. So the TF2D, the TF2B, the TF2E, and the H, and all these other factors bind, and they provide in a sense, a binding site for the RNA polymerase. So the RNA polymerase is not binding directly to the DNA like the E. coli RNA polymerase. It's actually binding to a protein complex. So we have the binding of these factors on there, and these things can sit there for a long time. And then what happens is, in order to initiate transcription, there is a phosphorylation event where the C-terminal region of one of the RNA polymerase II subunits gets phosphorylated, and that's shown by those two P's on the very bottom in the E section. Actually, it's a little bit misleading because there are 50 phosphorylation sites on the C-terminal domain of this subunit. And of course, there's lots of serines there. Serines have an OH. So there's all these serines at the C-terminal domain, and they all get phosphorylated. And what that phosphorylation does, presumably, is it completely changes the structure of the polymerase. It changes these charge interactions because now you have all these highly negative charges. And that is thought to be the initiating event that then allows the transcription to begin. And so the polymerase begins transcribing the RNA, synthesizing the new RNA molecule, which shows RNA transcription. You have that darker blue coming out that represents RNA, and then it goes down the line. Of course, you need the nucleotides, UTP, ATP, CTP, GTP. These are ribonucleotides. If they were deoxyribonucleotides, they'd have a D before each one, so like DATP, DCTP, DGTP, and of course it would be DTTP, not UTP. And then some of the factors come off, but some of the general transcription factors come off, but some of them stay on the polymerase and participate in stabilizing the polymerase, and also later at the end, termination and some of the other events that take place. So it goes along as like a cluster of proteins, large cluster of proteins that transcribe RNA. Now, I want to just say a few words about a protein called TF2H, which is one of the general transcription factors. So TF2H, we know what that protein does. It actually has two functions. TF2H is one of the general transcription factors. It does two things. It acts as a DNA helicase. And of course, we know from the DNA the DNA replication, which we talked about, that the DNA helicase unwinds DNA. So then it unwinds the DNA and allows the polymerization of the nucleotides. But it also has another function. It's a protein kinase. And this is thought to be the subunit or the general transcription factor that is activated to phosphorylate the RNA polymerase. So TF2H does at least two things: DNA helicase and DNA protein kinase, and a lot of these other proteins also have multiple functions, but a lot of them are just have not been worked out. Now, this is the establishment of the transcription initiation complex, large complex. These are called general transcription factors. They're called general transcription factors because these are the factors that bind to every single RNA polymerase II promoter. Because like I said, at least 95% of the promoters that are transcribed by RNA polymerase II have the TATA box. And so you get the binding of the TF2D and all these other factors. So these are general transcription factors. There's no distinction between what type of promoter is being activated. So they bind to all the promoters. So they're general. There's no specificity there. We'll look at specificity next time. So I talked about the TF2D binding and then other proteins binding to DNA that are required. And I want to just give you an example, tell you about some techniques, some experimental techniques that are used to identify proteins that bind to DNA. So you realize that in an experiment, in a system like this, where you have the TF2D and the TF2B and the TF2E and all these things binding, we know these by experiments that people have done. And it took years to figure this out, in the order by which they bind. And so I want to just give you just a slight view on how some of these things are done. I'm going to give you kind of a simple example. And it's done by what is called electrophoretic mobility shift assays. Electrophoretic mobility shift. And that's going to be described here. Okay, so the question at the top: how do we know, or how do we detect proteins that bind to DNA? So I'm going to describe to you electrophoretic mobility shift, and I'm going to describe to you how it can be used in two different ways. So, well, I'm going to tell you electrophoretic mobility shift. I'm also going to tell you something else called DNA affinity chromatography, which is a little bit separate. But the first thing is the electrophoretic mobility shift, and there's four parts, 4 elements to the system. The first is a piece of DNA, can be 75 to 100 base pairs in length, and that would be, let's say, a piece of DNA that has the binding site for the protein that you are interested in studying. So if you're interested in studying TF2D, then you need to have DNA that has that Tata box. And then you label it with a radioactive tracer. We use P32, radioactive phosphate, because there's phosphate on the DNA and it's easy to add to it. Then you need proteins. You can have purified TF2D, which isn't easy to get, but later I'll show you how you can do that. Or you can use, let's say, just a nuclear extract. Just a nuclear extract. So you can take cells, isolate the nuclei, and then get the proteins out and use that. Then there's a binding reaction. The binding reaction takes place in a test tube, small test tube, with various salts and magnesium that allows the right conditions, and the pH allows these things to bind. So what it is you take your DNA, you take your proteins, you put it into this binding reaction, you let it incubate for, it could be 20 minutes, half an hour, and then you take that mixture and you run it on by gel electrophoresis. Now, we looked at gel electrophoresis, and of course that is a way to separate, nucleic acids of different sizes, or to identify the size. And so I have on the left shown that diagram represents the electrophoretic gel, and then the migration goes from minus to plus, so it migrates downwards on the page. So if you took your piece of DNA and you had no extract, just the DNA alone, and you ran it on the gel, it would run pretty far down the gel because it's a little tiny piece of DNA. And then, of course, you can detect that by using either some kind of a digital or a digital detector, or a film, some way to detect where the radiation is. So you'd see a band at the bottom of the gel. Now, if you add the extract and you incubate it, and there's a protein that binds to it, then when you run that gel, what happens is the majority, depending on how much it binds, the majority of that band will have moved up in the gel as if it's larger. Towards the bottom, I show some unbound. That's that line towards the bottom. But the DNA that's bound gets shifted up. It looks like it's larger because there's a protein bound to it. You don't see the protein because the protein is not radioactively labeled. All you do is you see the shift in the band. So if you get a shift in the band up, it's because there's a protein bound. So how's that used? So this is on the right-hand side. So how is this used? It can be used, for example, to test the sequences that are required for binding of TF2D or Tata binding protein, let's say. So we want to identify what sequences does the TBP bind to. And you'll see later we'll be talking about all different kinds of DNA binding proteins, and this is how these kind of experiments are done. How do we identify what nucleic acid sequence does a protein bind to? So here's how it's done. You generate, in this case, I just made this up, 4 different DNA sequences. TAT, which is the classic Tata box. Then you make a change, TAC. You make 2 changes, or not a change in a different nucleotide, TATT, and then you make a bunch of changes in number 4, where you have a whole bunch of T's and 2 A's and a T. And you label those with radioactive marker, or the P32. And you mix those with, you can have, let's say if you have purified Tata binding protein, incubate it, run the gel. So here's what you see in the gel. And this is really pretty much what it would look like. So in the first lane, there's no extract. And that's why the band is at the bottom. In the second, 1 and 1, actually what it means is number one is at the top. In both those cases, we're using the same DNA. It's the TAT-AAAT, but in the first very first lane, there's no extract. So it runs fast. If you add the extract, which it is, you can see on the bottom I have which ones have nuclear extract added. You run the gel and you see there's a shift. It's running more slowly because the TATA binding protein, or TF2D, is bound to that. So you get a shift up. So okay, so we know, well, the TATA binding protein can in fact bind to the sequence. Then we, in number 2, which represents the second DNA sequence, we change the T to a C, run the same experiment, you see there's no binding. So that single change causes an inability of the TBP to bind to the DNA. Number 3, we change that fourth A to a T, and we get some binding. So it's a weaker binding. And number 4, we have a bunch of T's, and you see there's no binding. So what we say is the classic TATA box is the most effective binding site. If you make mutations or changes in the sequence, doesn't bind so well. And if you ever find a promoter that has a TATTAAT, you may think it's not a TATA box, but actually it may be. It may bind weaker, but it still might function. So that's one way to use this. Another way to use this type of experiment is to ask the question, what cell types is this protein expressed in? That's often extremely important to know which DNA binding protein is expressed in which cell type. And for this experiment, you would use the classic TATA box sequence and label it. And then you'd make extracts, nuclear extracts from different tissues, brain, kidney, skin, liver, so on. And then you would take that nuclear extract, bind it with the DNA, let it incubate, then run the gel. And what you see might be something like this. I just made this up, but that's kind of what it would look like. So you would see no binding in the brain, but binding in kidney, skin, liver, and colon, but not in muscle. So it's a way to identify what tissues a particular DNA binding protein might be in. Now, the last thing I want to show you is another technology or technique that can be used to purify these proteins. So once you know, for example, the binding site, so let's say we're talking about TF2D, once we know the binding site, then we can use that as a way to fish out the protein. Fish out the TF2D. So let's take a look at that. So what it says is, once the correct DNA binding sequence is established, you can purify the protein by a technique which is called DNA affinity chromatography. DNA affinity chromatography. So how does this thing work? So if you look in the very middle of the page there, I have this, it looks like a kind of a test tube, but it has a spout on the bottom. And these things can be very tiny, or they can be very big, depending on how much you want to purify. But the ones I've used are, let's say, the size of your thumb. Okay, that's about the size we're talking about. But there's a spout on the bottom, which would allow liquid to run through, and so you can open and close that spout. Some of these can be, much longer. They can be a foot long or longer. And you fill that column, that test tube in a sense, you fill that column with a resin which contains the bound DNA. You can covalently bind it by a chemical reaction which would bind the TATA box DNA to the resin. The resins would be about the size of a grain of sand. They're plastic. Around the size of a grain of sand, they're round. And there's ways to link the DNA to that. And so what I have there on the right hand side, I'm showing it's like I took one and I magnified it so you can see there's a bead with the DNA bound, and of course it's double-stranded DNA. And in reality, there's many sequences bound to each bead, not just one. And so you put all those beads into the column, and so what is basically happening there is that you have the column, size of a thumb, let's say, filled, like jam-packed, with this bound DNA. So you take that, and you take your nuclear extract. Let's say you're trying to purify TF2D. So you take the nuclear extract from a cell you know has TF2D, lots of the protein from the nuclei, and you put it onto the column, and you let it slowly, slowly, slowly flow through. And it comes out, material comes out the bottom. And you wash it with like a saline buffer. You pour a lot of saline, you let it wash through. What's happening is that the TF2D, or the TATA binding protein, is binding to these sites because there's zillions of sites in the column. And anything that doesn't bind is washed through, just washed through. So you continue to do that, continue to do that. You can monitor the protein that's coming out. You can measure how much protein is coming out. At the beginning, there's a lot of protein because everything just comes out. And then you keep washing it until nothing else is coming out. And the assumption is that nothing else is coming out, but there is protein bound to those beads. Then what you do is you take a very high salt buffer, which breaks the non-covalent bonds. These would be either ionic or they would be hydrogen bonds. So you put a high salt buffer through that, and that releases the proteins from the bound DNA, and whatever comes out, you collect in a tube. And you would call that, in this case, we'd call it TF2D. So that would be, you purified the protein. Now, quite often when you purify a protein, you want to see, okay, what size is it? So you run it out of protein gel. And when they ran the TF2D on the protein gel, it looked like what it looks like below. So that represents the protein gel from the plus, minus the plus, the direction of migration. And you heat the sample to break up any bonding, and you run the gel, and what they found, and this is actually the way it was discovered, they found that the TF2D was made up of all these different size proteins. Two of those proteins would be the TATA binding protein. And that's actually how the experiment was first done. And the TF2D stands for fraction 2, the D column or something. It's a number that represents something that had to do with the fractionation and the purification. And they just kept the name. And that's how they went at the very beginning, when I told you about the TF2D as many, many, many proteins in a complex. This is how it was discovered. Okay, so there you have it. So DNA binding assays and how these factors can be identified to bind the DNA, what sequences are required, and how to purify these things. Okay, so we've looked at a relatively simple E. coli promoter with the sigma factors, important to know. We've also then started looking at the eukaryotic RNA polymerase II promoters. We looked at the transcription initiation complex, which binds to the TATA box by general factors, and a lot of those. And then we're going to talk about more next time, other factors that are involved in RNA polymerase II gene expression. Okay, have a good rest of your day.